Okay, I think we'll go ahead and get started. Welcome everyone to this week's Medicine Grand Rounds. We have a guest speaker this week, who is Dr. Gary Wu. But before I turn the microphone over to Dr. Barbara Young, the Chair of Medicine, to introduce Dr. Wu, I wanted to remind people to, if they have questions during the course of, or that arise during, that they would like to ask Dr. Wu, please write them in the chat, and then I will ask those questions at the end of his presentation. So, Dr. Young?
Thank you, Conrad. Well, it gives me distinct pleasure to introduce Dr. Gary Wu to you. He received his MD degree from Northwestern University and completed his GI fellowship at the University of Michigan. He has since been a member of the faculty at Penn for 28 years, where he's the Ferdinand G. Weisbrod Professor of Gastroenterology. He's also the director of the Penn Center for Nutritional Science and Medicine, the co-director of the PennCHOP Microbiome program, and the co-director of the NIH-funded Center for Molecular Studies of Digestive Disease and Liver Diseases. He has been elected to both ASCI and AAP. Research in the Wu Lab currently focuses on the impact of diet on the gut microbiome and its relationship to several aspects of host metabolism, including nitrogen balance, intestinal oxygen regulation, and epithelial metabolism. It's my distinct pleasure to introduce Gary, and I know we have a wonderful talk ahead. Thank you, Gary.
Great, thank you so much, Barbara. First of all, for the invitation to be out here. I wish I could be there in person, but I guess this is the next best thing. So what I'd like to do is talk to you about the role of human subject research in the microbiome and the notion of intersubject variability and how this is actually important in personalization of the response. And I'm going to really focus on diet because this is actually how we got into the microbiome space. But not only challenges, but the opportunities that this personalization and intersubject variability allow in the field. So these are my disclosures. I don't really have anything relevant to disclose this talk. So the human microbiome is exceedingly complex. It's not just bacteria, but there are viruses, archaea, microeukaryotes, other types of microbes that live on our body surface, and each site is actually very different from each other. But the gut microbiota is particularly unique because of its high biomass density of organisms and extreme level of complexity. Now, from a 30,000-foot view, this is how we interact with the gut microbiota. The gut microbiota exists in the external world topologically in the lumen of the gut, and the internal world contains the mucosal immune system. What serves as a barrier is the intestinal epithelium, but also in the colon, there's a sterile layer of mucus that completely separates us from the microbiota. But we still interact with each other. Things that we produce like mucus and antimicrobial peptides end up in the lumen of the gut that interact with the microbiota, changing its function and structure. In return, the microbiota actually interacts with us. Certain types of microbes, as well as their products, lead to an anti-inflammatory response. Other microbes are more pro-inflammatory. And bacteria in general can help program plasma cells to secrete specific immunoglobulin A's, which are, again, secreted into the gut lumen to help fortify barrier function. For the purposes of my talk, it's actually important to recognize that what you eat in terms of diet, antibiotics, and xenobiotics not only shape the composition of the microbiota, but diet in particular is a substrate for the microbiota to produce a whole myriad of small molecules or metabolites that we normally are not able to produce. We absorb those molecules, and together with its effects on mucosal immune responses, we think that the gut microbiota may be playing a role in the pathogenesis of many different types of diseases, principally because a lot of diseases have in common the notion that intestinal inflammation or inflammation in general is actually important. So again, we got into this area of the microbiome thinking about diet. And I just want to share with you this one slide about mice versus humans and diet in the microbiome. So there are challenges to characterizing the effects of diet on the human microbiome. Humans are poorly adherent to standardized dietary regimens. Current tools to characterize dietary composition and intake are imperfect. We basically use dietary surveys. There's a reciprocal nature of dietary composition to maintain isocaloric consumption, making it difficult to determine what factors are responsible for an observed outcome. If you change one thing in diet, you actually have to change something else. Diet can have profound effects on the host biology independent of the microbiota. And both intensive controlled feeding experiments and large outpatient cohort studies are expensive and challenging to complete. And it is for that reason that molecular biologists like myself study animal models. And there's a lot of utility in animal models in studying the interaction between diet and the gut microbiota. You have tight control of defined diets over long periods of time. Multiple biological replicates are feasible. Germ-free animals can be used to examine the effect of diet independent of the gut microbiome. Defined microbial consortia and complete human microbiota studies can be actually performed in notobotic mice. And most importantly, you can determine cause and effect relationships involving diet and the gut microbiota. But a really fundamental question is, are mice the same as humans? This is one of the very first studies that we published together. We did this with Rick Bushman, who's now the chair of microbiology at Penn. And so in this study, we examined two different types of mice, a wild-type mouse and a knockout mouse. And essentially, we put them on two different diets, a normal diet and a high-fat diet. And you can see just by looking at the structure of the microbiota, the histograms here look very different on a high-fat diet than on a normal diet, irrespective of the genotype of the mouse, either being wild type or knockout. So another way of looking at this, it's a principal coordinate analysis. So each dot here is a different fecal pellet from a mouse, and it's the entire consortium of that composition of that microbiota boiled down to one dot. Dots that are closer together are more similar to each other in terms of microbiota composition, the dots that are further apart. And so what I'm essentially showing you here on the first principal coordinate analysis, principal coordinate, is that 28% of the variance can be explained by diet, whereas only 6% of the variance can be explained by genotype, meaning that diet is the primary driver in the composition of the microbiota, not the genotype of the mouse. So that's what we observe in mice, very consistent alterations in the microbiota and very large effect sizes. What do we actually see in humans? So this is a human subject study where we did something actually similar in humans. We had 10 healthy volunteers. We randomized into a high-fat versus low-fat diet. And then we collected fecal samples every day for 10 days. And what I'm showing you down here is another one of these principal coordinate analyses. Each individual is a different color. Each ball is a different fecal sample or a different day of the study. And the first thing you notice is that all the colors are separate from each other. So this is intersubject variability. So this means that everybody's microbiota is actually very different from each other. If you notice, and you look carefully, for each color ball, there's always one ball that's different from all the other balls of the same color, and that's what I've drawn an arrow to. So that one ball that's different from all the other balls of the same color is day one of the study. So that essentially means that the human microbiota changes within 24 hours of eating a different diet. But the important point here is that the impact, although highly statistically significant from day one to all the other days, is relatively modest in comparison to how different we are from each other. The other point is that the vector moving from day one to all the other days is seemingly stochastic. It's not consistent, even though individuals were on similar diets. And a corollary of that is that people didn't converge in terms of their composition of microbiota over 10 days, even though they were on defined diets. This observation in intersubject variability and the response to diet was actually also observed in a more recent paper by Dan Knights, where he studied 34 people, collected a lot of dietary records, did a lot of sequencing, and he came to the conclusion that similar foods have different effects on different people's microbiomes. So this is the notion of intersubject variability. Some people would say, well, this is noise. This is really difficult to deal with because people are so different from each other, and you can't predict which way people's microbiomes are actually going to move. But I would say that that's actually information that can actually be utilized through high dimensional analyses to make personalized predictions. And so this is a seminal paper by Aaron Segal and the Weizmann Institute, where they noticed that the postprandial glycemic response in individuals eating the same food was highly personalized, high degree of intersubject variability. So if you match that intersubject variability and postprandial glycemic responses with the high level of intersubject variability in the microbiome, together with some other ancillary information, you can come up with a computational algorithm that actually predicts what type of foods an in particular individual should eat to minimize the postprandial glycemic response, with the ultimate goal of coming up with better diets for the treatment of type 2 diabetes. So these types of observations have been supported by subsequent studies. It's a little bit early right now. We don't know how well this is going to work, but it is a notion that intersubject variability is not noise, but it's actually information that can be utilized for personalization. So everything that I've shown you previously really builds upon this notion that I really like, and that is collaborative science between basic science research and clinical investigation. So I'm a wet bench basic science researcher. What we spend a lot of time doing is develop model systems that phenocopy the human response. And we use different types of systems to actually do this. In the clinical world, there's epidemiology, clinical phenotyping, and we study biospecimens. But the studies are actually bidirectional, because the way that we do science over the past couple of decades is fundamentally different now, because we actually have these high dimensional analytic technologies, genomics, metabolomics, as well as computational and bioinformatic algorithms that allow us to take a more systems biology to really understand the complexity by which either animal models or humans actually respond to the microbiota or to diet or other environmental influences. The other point about this is I always thought about research as being bench to bedside, but there's another way of thinking about the research, and that's actually from the bedside to the bench, where you actually study human biology, make correlations, and then use that as a way to generate hypotheses that can actually be tested in animal model systems or at the wet bench to prove cause and effect relationships. Because you may not understand why there could be certain correlations in humans, but by doing the basic research, you will understand those biological mechanisms. But that's a shortcut because you already know that those associations and correlations already exist in humans. So working bidirectionally in science, I think it's a wonderful opportunity. When you do this type of research, ultimately, you get into human subject research, and this is where we think about new modalities to prevent and treat disease. There are a number of things that are actually important when you think about doing human subject research. I won't get into details here, but it's about collection of biospecimens, collecting and deeply phenotyping your human subjects in your studies. So with that as an introduction, there are three things I want to cover. I want to talk about diet, the microbiome, and IBD. I want to talk about personalization of the gut microbiota, and I'll make a parallel with Darwin's theory of natural selection. And then I want to talk about culturally distinctive characteristics of the microbiota and diet. So we know that IBD is very complex. It's part genetic, part environmental, and that includes the microbiota, together with an immunoregulatory defect that leads to chronic inflammation. We believe that the genetic contribution to the development of Crohn's disease is estimated to be about 30 to 40%, and only 10% in ulcerative colitis based on the concordance of IBD and identical twins. So it suggests that environmental factors are the largest contributor to the pathogenesis of inflammatory bowel disease. And an example of this is the increasing incidence of inflammatory bowel disease that we see that began in Western civilization about 150 years ago, but now we're beginning to see on a rapid rise with industrialization in Asia and India and China. So these are very powerful environmental effects that basically demonstrate the important role that the environment plays in the pathogenesis of various types of inflammatory immune-mediated disorders. Now, we do believe that diet is one of those environmental factors, and for decades, investigators have been studying therapeutic diets for the treatment of inflammatory bowel disease. David Susskind here at Children's Hospital is a leader in this field. And when you think about diet, investigators have been looking at things that you can add to diet, things you could exclude from diet, or things that you may modify in terms of diet for the treatment of inflammatory bowel disease. So just starting with things that you can add to the diet, I want to bring up the notion that we live in a mutualistic relationship with our microbiota. Intestinal mucus provides a source of nutrition for the microbiota, and in return, the microbiota digests undigestible carbohydrates and leads to the production of really important small molecules like short-chain fatty acids. And the reason the gut microbiota is so actually good at doing this is it contains a lot of genes that our genome actually does not contain that can digest these complex glycans, carbohydrates, leading to production of small molecules like short-chain fatty acids that again regulate the immune system and then also play a role in fortifying barrier function through the colonic epithelium. But there's another way to think about dietary fiber that may actually be beneficial in maintaining barrier function besides just short-chain fatty acids. So this is work by Eric Martens. So what he showed in an autobiotic animal model system is that if you feed these mice a fiber-rich diet, the microbiota digests the fiber that's actually in diet and it leaves the mucus intact. But if you feed these mice a fiber-free diet, the microbiota will now turn its attention to actually digesting the carbohydrates and mucus, depleting mucus, destroying the barrier function, leading to inflammation. Another way to think about diet therapeutics for IBD is using a modified diet, and probably the best defined modified diet for inflammatory bowel disease are these elemental or defined formula diets. Many of these are liquid formulations that need to be delivered by nasogastric tube, although some can actually be consumed orally. And there are many studies that show that they can have significant efficacy in the treatment of Crohn's disease, but we don't really understand how they actually work. So with that notion in mind, what I want to do is tell you about a study called Food and Resulting Microbial Metabolites, where we actually did study those defined formula diets. The objective was to determine a relationship between dietary composition, the composition of microbiome, and metabolite products that are all ultimately present in the gut lumen and the plasma of humans. Our strategy was to use 3 divergent diets: vegans, omnivores, and omnivores randomized to consume that exclusive enteronutrition diet used to treat inflammatory bowel disease, and look at their impacts on the composition of the gut microbiome as well as the metabolome. And I'm going to just, this is a spoiler alert, this is actually what we found, but I'm going to tell you up front. This was not anticipated, but what we discovered was that dietary fiber was the primary driver of all the observations that I'll show you. And the effect was so large that we saw a very consistent effect even though we were only studying a very small number of individuals, that actually exceeded that intersubject variability, which is often a challenge in human subject studies. So this is the design of the study: 10 omnivores, 10 consuming enteronutrition, and 10 on a vegan diet. And what I'm showing you here in the first 5 days is a dietary phase. What happened to the microbiota on these different diets? Halfway through, we actually reduced bacterial load by giving antibiotics and a bowel purge. And what you can see, the dark blue bars here are basically our sequencing reads that are microbial in origin. When you reduce bacterial load in the gut, you can only really see the host reads, the human reads, because you largely depleted the gut microbiota. But then very quickly, the microbiota actually recovers. It reconstitutes itself. So by looking at each one of these phases, mapping the composition of the microbiome to the metabolites, we can actually infer functionality. So this is just a point that looking at fiber, vegans in green here consume a lot more dietary fiber. But people on an enteronutrition diet basically are not consuming fiber. So what happens on these various types of diets? Shannon diversity is looking at the number of different types of microbes in an environment and their proportion. an omnivore, vegan, enteronutrition diet. Remember, the enteronutrition diet, they actually started off as omnivores until we randomized them. They all have high levels of diversity. It drops down when you clean out the gut, but it then begins to recover. But on an enteronutrition diet, it doesn't actually recover. So what happened to the structure of the microbiota? So each day of the study is color-coded. Each symbol is a different dietary group. During the dietary phase, everybody's over here. Each stool sample is a separate ball. And what I can show you is that within 5 days, these individuals on that exclusive enteronutrition diet are now down at the bottom of these ellipses, whereas all the vegans and omnivores at day 5 are all clustered up here together. So vegans and omnivores, even though they eat very different things, their composition of the microbiome is actually very similar to each other. But EEN, enteronutrition, has a very extreme effect that exceeds the difference between a vegan and an omnivore. During the recovery phase, the vegans and omnivores over here on an enteronutrition diet, you're now completely separate from everybody else. So that's the structure of the microbiota. Why are they so different from each other? So here we actually look at the genetic composition of the microbiota, focus on glycoside hydrolases. These are those enzymes that I mentioned previously that are important for the microbiota to digest fiber and complex carbohydrates. So I'm showing you here all the different genes during the dietary phase that are actually decrease in enteronutrition diet. So red means it's increased, blue means it actually decreases. So during the first 5 days, glycoside hydrolases that are important in digesting plant products are actually reduced on the EEN diet because it goes from red to blue, but it's preserved on an omnivore and vegan diet. And that makes sense because these individuals are no longer consuming fiber. What they are consuming, they are simple carbohydrates like sucrose, which is this form of carbohydrate in these exclusive enteronutrition diets. And you can see glycoside hydrolases involved in simple carbohydrate metabolism have now gone from blue to red because they're now increasing. Now, we didn't actually notice that much of a difference in those types of glycoside hydrolases digest mucus, unlike animal model systems. But this may be just because we didn't treat people long enough, or maybe it's a difference between humans and mice. During the recovery phase, what we noticed are that glycoside hydrolases actually began to return on an omnivore and vegan diet because you're going from blue to red during the recovery phase, but they don't largely return on an exclusive enteronutrition diet, again, because they're not eating fiber. Yet the carbohydrates, the simple carbohydrate glycoside hydrolases are returning because you're going from blue to red here. And again, not that much actually changed with mucus. So that's the composition of the microbiota, suggesting that dietary fiber is the primary driver of this. But what about metabolites in the stool? And because I told you that what you eat not only changes the composition of microbiota, but actually plays a role in the metabolites in stool. So here we're looking at the metabolome of each fecal sample across the different days of the study. And these dotted lines indicate the enteronutrition diet in blue, the omnivore diet in red, and the vegan diet in green. And during the first 5 days, vegans and omnivores, the metabolites in stool are largely the same, but within 5 days, just like the composition of the microbiota, the metabolites in stool are now different than in day 0. In day 9, they shift away, but by day 15, if you're vegan and omnivore, you're beginning to recover and circle back around to where you were before. But the metabolome and the feces of individuals on an exclusive enteronutrition diet are now completely separate. So now we have 800 different metabolites, 3 different diets, and 4 different time intervals. And how are we going to understand all of that information? So a lot of time we spend doing a microbiome analysis is dimension reductionality, trying to reduce the dimensionality of all the data so we can actually wrap our heads around and understand what's happening. So our biostatisticians came up with a very simple algorithm that if the log ratio of a particular metabolite from one point to another is greater than 0, meaning it's increasing, we assign that a code of 1. If it's not statistically significant between 2 time points, it's coded as a 2. Or if the log ratio was less than 0, meaning it's decreasing, we code that as a 3. So each different interval here in the study can now receive a zip code. And so you have a 4-digit zip code that tells you for each metabolite the entire pattern throughout the entire study. And what I'm showing you here are all the different 4-digit zip codes and a pattern of all the different metabolites assigned an EEN diet, a Western diet, or vegan diet. So the first thing you can see that the zip codes for all the vegans are in the middle here. They're not on either end. And why is that? Well, it's because all these codes begin with 2, meaning that diet did not change the metabolites in the stool. So what I didn't tell you was that even though the omnivores were as locked up as an inpatient during the 2 weeks of the study, the vegans, because they're much more compliant with diet, were allowed to be outpatients. So they were eating their own diets at home. And it made total sense to us that their diet would not have an impact on their metabolome, which is why we don't see any difference here. They're all coded with a 2, meaning it doesn't change. The other thing is that most things did not change. They were coded all the way across as a 2, which is good. But we did see that metabolites did change on an EEN diet, but they only decreased. We did not see any increase in any metabolites. So this could be consistent with the notion that exclusive enteronutrition is excluding something from a whole food diet that could potentially be bad in patients with inflammatory bowel disease. So why these metabolites are important? Well, this is just a notion that metabolites have a lot of effects on us. They can serve as ligands for receptors. They can activate certain immune modulating pathways. They can serve as antibiotics. There's a lot of interest in amino acids and how they're converted into various types of metabolites. The pathways important in the conversion of, for example, tryptophan, aromatic amino acid, to endopropionic acid by the microbiota is something I'll highlight on the next page here. Endopropionic acid is actually a ligand for the nuclear hormone receptor, PXR. And PXR has been shown to fortify barrier function in the intestinal epithelium. And PXR is a susceptibility locus for inflammatory bowel disease. So these small molecules may be fundamentally important. So I'm showing you in our study the pathway from tryptophan to endopropionic acid. And looking at tryptophan levels in plasma and stool, the first thing you'll notice that is upon recovery, tryptophan levels in the stool during the recovery phase on that EEN diet, which is blue, is actually higher than a vegan and an omnivore. Yet the opposite is true when we think about the product endopropionic acid. Upon the recovery phase, endopropionic acid made by the microbiota is actually much lower on that exclusive enteronutrition diet. The reason for this is that the microbiota is now different on that exclusive enteronutrition diet and no longer contains the genes important in the conversion of tryptophan to endopropionic acid. And because you can no longer make that conversion, we think that the substrate, tryptophan, builds up at higher levels in the stool. So it's the inability to make this conversion that leads to higher levels in the stool. We know this because we looked at the different types of organisms, the firmicute organisms that are responsible for making that conversion. And if we look during at the recovery phase in an omnivore vegan diet, that there's high representation of these organisms as the microbiota recovers on an omnivore or vegan diet, but very much less so on an enteronutrition diet. So these organisms are not recovering. And for that reason, we don't see that conversion and the development of that metabolite. So that's just one metabolite. But what if we look at all the different amino acids and we look at all the amino acid derived metabolites? So this is what we did here. And we looked at the purge phase when we're flushing out the gut, and upon reconstitution that the microbiota actually recovers. And what I'm showing you is all the different types of amino acids. When it's coded as a green, that means it's increasing during that purge phase. And when it's orange, that means it's actually decreasing during that reconstitution phase. And you can see that that happens during the purge phase. Amino acid levels are higher in the stool. And we think that's because you're flushing out the gut of these amino acids before they can actually be absorbed by the host. But Upon recovery of the microbiota, you can see that these amino acids begin to decrease in stool. But you actually don't see that on the enteronutrition diet. Those amino acids don't seem to decrease. So why is that? Well, if we look at amino acid metabolites, it's exactly the reciprocal pattern. Whereas, in fact, as amino acids build up in the stool, they're actually, their derivatives are actually decreasing. And we believe that's because the microbiota is no longer there to make the conversion of these amino acids into these metabolites. But as the microbiota actually recovers, those amino acid metabolites begin to appear again, and then amino acid levels drop. But you don't actually see that on the EEN diet. And that's essentially what we showed in the indopropionic acid, but we can actually see that in other amino acid derivatives that have been previously defined as other types of metabolites like cadaverine that's made by the microbiota. But the interesting thing about this is that by looking at pattern matching, we identified other types of amino acid metabolites that have never been previously associated with the human gut microbiota. And that means that perhaps there are new types of metabolites that could have functionality. So in summary, the gut microbiota on a fiber-free diet differs from that of an omnivore or vegan. The lack of dietary fiber slows microbiome recovery after an ecological stress. These dietary effects on firmicutes affect both carbohydrate and amino acid metabolism. And I didn't mention this, but in fact, what we saw here was that the effect of the metabolites in stool had a very small effect on plasma metabolites. So most of the plasma metabolites, I think, are from diet, with a small number are actually from the stool and from the microbiota. So from a clinical standpoint, why is this actually relevant? Well, first of all, we think about the production of metabolites through the gut microbiome in a very linear fashion. You take a particular dietary constituent, the microbiota converts it into a particular carbohydrate-based metabolite like short-chain fatty acids. What I'm saying is that in certain circumstances, dietary carbohydrates, and this would be potentially if you're on IBD and you're on an enteronutrition diet, or you're a patient in the intensive care unit, not eating food, but on antibiotics, you may disrupt your microbiota in a way that dietary fiber now is important in helping you to preserve the functionality of converting amino acids into amino acid metabolites. So it's not only linear, but the dietary fiber could have an effect on the microbiota in a completely different class of dietary molecules, amino acids. And that's added functionality that may be important for the host. And we're actually very interested in doing studies in human subjects, sort of building upon this notion. Again, the whole point is that investigators are now beginning to focus a lot on thinking about mining the microbiota for small molecules, in part through diet. That could be the next set of small molecule drugs because they interact with receptors in the body. So these are bioactive molecules because organisms spend a lot of time and energy making them. And this is an opportunity to think about new drug discovery through the microbiota. All right, so what's another view on how these diets could be beneficial in the treatment of inflammatory bowel disease? Well, maybe they're excluding something that's actually bad in our diet because our diets are now westernized. And there's a big interest now in ultra-processed diets and how this might be actually associated with various types of diseases. And there are studies in animal model systems showing that artificial sweeteners and dietary emulsifiers will change the composition of the microbiota and lead to an inflammatory response in mice. The relevance of this to human biology is not well understood, but we actually just completed a study where it was a randomized control study feeding healthy human subjects dietary emulsifiers to determine whether or not things that were actually observed in animal model systems is actually reproducible in humans. And so we're analyzing that data and hope to be describing the results in the near future. But at the end of the day, There are diets that are being developed. I'm showing here the Crohn's exclusion, Crohn's disease exclusion diet, largely getting rid of things that you see in processed food and adding whole fruits and vegetables. And as I mentioned, David has done a lot of work at Children's Hospital focused on the specific carbohydrate diet. Okay, so let me move on to personalization of the microbiota and the notion of natural selection. So we notice in IBD, the microbiota is actually very different than in health. It's dysbiotic. It's different. And why is it different? Well, it's because it's a complex microbial community responding to environmental stress. And that environmental stress in IBD is inflammation. So inflammation leads to an oxidative environment. It produces small molecules that favors the outgrowth of certain types of organisms. So we cause that dysbiosis, but does that dysbiosis play a role in disease pathogenesis? Well, in animal models, we think that it does, because you have animal models that are genetically engineered to develop colitis. And if you transfer their microbiota into a healthy mouse, under certain circumstances, they'll actually develop colitis. So that transferability suggests that that altered composition of the microbiota may be important in disease pathogenesis. With that notion in mind, if disease is associated with an alteration in the microbiota, maybe the strategy is to change that dysbiosis by one of many different means to actually promote health. And what I'll focus on is fecal transplantation. As you all know, fecal transplantation is highly effective in the treatment of an infectious disease, Clostridioides difficile infection. That's a growing problem. And it's because antibiotic use leads to the outgrowth of an organism that produces a toxin. About 20% of these individuals are refractory to treatment or have recurrent disease. But remarkably, simply taking fecal material from a healthy individual and transplanting it to an individual with disease is 90% effective in the treatment of recurrent Clostridioides difficile infection. With the notion that it could treat an infectious disease, other investigators have now begun to explore whether or not you could treat more immunologically based chronic disease processes like inflammatory bowel disease. So there are 3 placebo-controlled trials in small numbers of individuals showing that fecal transplantation may have lead to a significant improvement in ulcerative colitis. This is early days. I caution people that this is still experimental. But it is a window into human biology that, if validated, it could lead to important observations. One of these observations, it was in this study, that 7 of the 9 patients that developed a remission after FMT received fecal material from a single donor. So there's always been sort of this interest, and maybe there could be a super donor for FMT that has a particularly good composition of their microbiota, maybe because of their genetics, maybe because of their diet or their immune response, that they have a composition that's particularly good for the treatment of a disease. And one of these notions is that you would have high levels of Clostridia taxa, and these are the same organisms that produce short-chain fatty acids. So there could be a personalized response in thinking about that donor. Again, all hypothetical right now, but there are a lot of people interested in this notion. But there is a personalization aspect to fecal transplantation. And this was shown by Eric Alm, where he studied 4 donors into 19 patients, did a lot of sequencing and very sophisticated bioinformatics. And what we found was that there are certain individuals that will actually engraft organisms from the donor. But that engraftment, what organisms actually engrafted, were actually very personalized. It's not the same in every individual. And you could actually make predictions as to what organisms would engraft after fecal transplantation, largely based on what was already there in the recipient before they actually got transplanted. So that's my notion of personalized engraftment of organisms. And this extends to probiotics. This is work by Jens Walters, where he looked at individuals getting a probiotic, a bifidobacterium organism, and only 30% of those individuals actually engrafted that organism. So that engraftment was actually very individualized, and it had a lot to do with whether or not you already had your own bifidobacterium already there. This observation was also followed up by another very interesting study. Again, the notion that the engraftment of these organisms may be actually very personalized. And it may be that that's why probiotics don't have a bigger effect, because their effects may be dependent upon whether or not they're able to engraft. And this is based on a very personalized type of mechanism. So why are this engraftment very personalized? This is where I'm talking about Gauss's law and Darwin's theory of natural selection. The notion is that complete competitors cannot coexist in the same niche. So when one species has even the slightest advantage over another, the one with advantage will dominate in the long term. So think about these yellow birds picking off and eating bugs on the trunk of a tree. If another larger bird comes along that wants to eat the same type of insects here, the yellow birds get displaced. Either they're going to become extinct or they have to go and look for food someplace else. And that's a notion of a competitive niche exclusion. And we can show that in bacteria, and this was done by Sarkis Mazmanian. If you engraft a certain organism in the gut of a mouse, and then you use a similar organism, you try to engraft it, it won't actually take hold because it extinguishes over time. And the first organism that engrafts is actually the dominant winner. And the reason for this, at least in this model system, are specific genes in the genome of the bacteria that are important in carbohydrate metabolism. So why do I mention this to you? In my last slide, I'll show you that the world is changing the microbiome field. We're moving on from stool to live biotherapeutic products, and people are making products now in the pipeline to treat disease using defined microbial consortia. And so this is work that was published in Gastroenterology, where this company treated patients with ulcerative colitis with defined organisms. And what they found was the engraftment of these organisms, and the response was most significant in those individuals that received vancomycin, an antibiotic, as pre-treatment. And that is consistent with the notion that you may need to open up the niche that allows the engraftment of specific organisms to get more meaningful results. All right, so the last thing I'll talk about is the culturally distinctive characteristics of the microbiota and diet. As I've been saying all along, diet not only changes the composition of microbiota, but leads to the production of small molecules that are actually important. So a number of years ago, we studied vegans and omnivores and looked at their plasma metabolome, metabolites in blood of vegans and omnivores, and looking at different types of metabolites here. And if it's positively correlated with vegans, it's red and associated with green. If it's negatively correlated with omnivores, in purple here, it's blue. And so you can basically see for any particular metabolite, it's almost reciprocally related. Things are positively correlated with vegans are negatively correlated with omnivores and vice versa. In fact, the plasma metabolome of vegans and omnivores are so different that by using random forest analysis, a machine learning algorithm that stratifies by importance, features that predict a dichotomous outcome, either a vegan or an omnivore, we can predict with 94% accuracy if you're a vegan or omnivore simply looking at 30 metabolites in blood. The ones that are from the microbiota I've drawn an arrow to. So the point is that most of the metabolites that are most important in the difference between a vegan and an omnivore are not from the microbiota, yet the microbiota does play a role. And if you're a vegan, more of your plasma metabolites are from diet and your microbiota than if you're an omnivore. And the reason for that is that vegans consume more plant products, and the microbiota is converting these plant products into small molecules that we can actually measure in blood. But it's more complicated than that. So this is something that Joanna Lampe is a world's leader on here at Fred Hutch, who studies Equal, which is non-steroidal estrogen that has possible health benefits. The interesting thing about Equal, which is actually produced from soy that you actually consume in diet, is that only 30% of adults in Western populations can produce Equal from soy, but double that amount actually are able to produce that in Asia. So in our omnivore vegan study, our omnivores were not eating much soy, so they weren't consuming a lot of the soy substrate, and they basically did not produce much Equal. But the vegans all consumed a lot of equal, and they were consumed a lot of soy, this Dadson precursor, but only about 40% of the vegans, even though most of them were consuming a lot of soy, only 40% had actually detectable equal levels. So the percentage of vegans that are equal producers didn't reflect diet. It actually reflected more the fact that they were living in the Western civilization than in Asia. And that's something that we actually find in multiple studies, that individuals living in different geographic locations, particularly in more rural locations and more industrialized nations, will have a very different microbiota. So in rural Africa and Burkina Faso, the childhood microbiota is actually very different than we actually observe in Europe. Part of this could be dietary fiber. So this is work by Justin Sonnenberg. And basically what he did is he studied mice and their microbiota in 3 different generations on a high fiber diet. And the microbiota is largely preserved from generation to generation. But on a low fiber diet, from one generation to another, you begin to lose more and more microbes. And even within that same generation, if you then begin to give a high fiber diet, you could only recover a certain proportion of the organisms, never getting back to high diversity with all those organisms. And the notion is that with industrialization, we're beginning to lose components of our microbiota over time. That the fear is that we'll lose them permanently. And these may be important and try to ultimately prevent disease. This is in mice, but Dan Knights did this very interesting immigration study where he looked at the immigration of individuals from Southeast Asia and looked at the composition of the microbiota before they emigrated and then after they emigrated, and whether or not they were short or long-term residents, and looking at subsequent generations. And what he essentially found was that Prevotella strains, which are actually very predominant in more agrarian cultures, you begin to lose these organisms after immigration and subsequent generations, and you replace them with Bacteroides strains. And so that's the notion that this could be somehow tied with health in some way, but it's actually important to know that diet only explained about 16% of the total variation. So it's not the only thing that's leading to this change in the microbiota. It's something else in the environment that is actually important. But the inference is that, well, maybe this change could be playing a role in metabolic syndrome and diabetes and obesity. But I want to show you, just to keep it honest, there are challenges in translating these things into human biology. There's a very important paper by Ellen Black, where she took adults that were obese with metabolic syndrome, and she treated them for 10 days with vancomycin and amoxicillin. And in this study, a whole bunch of metabolites associated with metabolism, metabolic function of the hosts were changed, short-chain fatty acids, bile acids changed, the composition of the microbiota changed dramatically. But when they did deep metabolic profiling of the human subjects, there was no difference in metabolism in these individuals, at least in the 10 days of this study. The other point is that, getting back to fecal transplantation, there have now been a number of studies looking at fecal transplantation and metabolic syndrome and obesity, with the hopes that there might be a signal for improvement if you change somebody's microbiota. And there are some studies that show modest improvements in hemoglobin A1C and cholesterol, but the impact is modest. There have been no studies that show, compared to placebo, that there are any effects on obesity. The quality evidence is still low, and more studies are actually needed. So I don't mean this to be negative. I'm just saying that there's a lot more to know and to learn. And this is the importance of doing human subject research in the microbiome space, to think about translation. That's ultimately going to be important. So there's a bidirectionality of microbiome investigation, where, again, people like me study animal model systems to find environmental conditions, to find genetics, monotonous diet. And for this reason, there's a very high signal to noise ratio that allows us to see things and observe things and allow us to determine proof of concept cause and effect relationships in a modest-sized cohort. On the other hand, humans are much different. They're free living in a variable environment, genetic diversity, variable diet, and there's a lot of noise. Signal to noise ratio is much lower because of intersubject variability. But the important thing to keep in mind is that small effect sizes over large populations can be still highly impactful, and that intersubject variability is not noise. It's actually something that can be embraced for personalization and thinking about therapeutics involving the gut microbiota. So my last slide is diet can have a direct effect on the host. Diet could have an effect through the microbiota. The type of diet may be important. It has an effect on on bacteria. I didn't talk about its effects on other types of organisms, but there's a literature on that. The effect on the microbiota through diet is not only on composition, but also on metabolic products. And there are a lot of people thinking about medical foods, the next generation of prebiotics, substrates for the microbiota, next generation of live organisms, probiotics, and a combination of both of them for the prevention and or treatment of disease that are different than the ones that are currently on the market. And we're now in the era where we believe, at least for Clostridioides difficile infection, in the near future, we will actually have live biotherapeutic products that will get us beyond just using stool for the treatment of that one disease. But there's a lot of interest in combining science and technology, clinical practice, regulatory aspects, along with intellectual property and business opportunities. Many companies out there developing strategies for the treatment of disease or prevention of disease through the microbiota that make this a very, very exciting time for microbiome research, acknowledging the fact that we currently don't know everything, that there's much more work that's actually needed. So with that, I want to acknowledge, I spent a lot of time talking about the farm study. I'm one of a group of many investigators that work together, and none of this could be done without a group effort, emphasizing the need for multidisciplinary team research, and this is our sources of funding here. So with that, thank you very much, and I'd be delighted to answer any questions.
Thank you, Gary. I think Conrad is moderating, but I'm happy to jump in.
Oh, no, I'm sorry. I was on mute. I'm sorry about that, Gary. I would say thank you very much, Gary, for that comprehensive and insightful presentation on the gut microbiome. A few questions have come in. We'll start off with number one. Number one, it seems like the diet doesn't have that much effect on the microbiota and metabolites until after antibiotic administration, the purge phase. If someone is choosing to change their diet, should they undergo the purge phase before? It's almost like a conditioning for a transplant, right?
Yeah. So no, I don't think people should. This was a human subject study, and it was under IRB protocol, and we monitored people very carefully. There were no adverse outcomes. In fact, when we saw the extent to which we changed the microbiota, which was actually unanticipated, we actually modified our IRB and actually called back these individuals to follow them up and collect subsequent stool samples for sequencing just to make sure the microbiota had actually returned from a safety standpoint. So I would not recommend that people take antibiotics for this. You know, it depends on the depth that you're looking at things. There are changes that actually occur in the metabolome and the microbiome with diet. There's no question about that. And there are even really interesting studies on circadian rhythm. The timing of your diet will have an effect. But my point is that there's a large level of individuality in what you produce in the composition, which means that it's not the same for everybody.
All right. A second question is, can you talk more about how infancy or the microbiome during infancy or as a child sets up individuals for health or disease in later life?
Yeah, this is something that's of significant interest to us. We actually run an infant growth and microbiome study at Children's Hospital where we track individuals, our infants from birth out to 3 years of age. So there's epidemiologic data suggesting that the microbiota and the use of antibiotics are associated with development of obesity and diabetes. And so there's a lot of interest in this field. And animal model systems, you can actually show that early life events are actually very important. I think that there's growing evidence that this may actually be true in humans. I think allergic responses, atopic disease is a really good example of this. So the reason why this is so important is that the mucosal immune system is actually being developed early on in life, and it's very sensitive to the environment. So it is very plausible based on atopic disease observations that early life events may be important in predisposing to development of atopic disease, because there are actually some really good studies in childhood in where they actually live and their protection or their predisposition for developing atopic disease. This may relate to other types of diseases, but that's a really active area of research right now.
Another question, is there any benefit for so-called commercial probiotics after antibiotic treatment? What should be in the probiotics if somebody is going to take a probiotic after they take antibiotics to restore their normal gut?
Yeah, the evidence for this is unfortunately poor right now. So if you're interested, if you go to Gastroenterology or just Google probiotics and the journal Gastroenterology, there was actually a technical review and a clinical guideline recently produced by the American Gastroenterological Association on this topic. Largely, the evidence is very small and to non-existent that these things provide significant benefit in the treatment or prevention of any particular disease, and that's why they're not used as drugs. My notion is that, if this is a physician that's asking this question, as a physician, there are patients that absolutely swear by probiotics, that they have discomfort and they feel better. And I can't deny the fact that it's providing them some benefit, but it may be in a very personalized way. The other point is that, probiotics that are currently on the market were not designed to treat a particular disease. They were designed to actually make fermented milk products like cheese and yogurt. So that's the opportunity. Use our current knowledge in genomics and microbial biology to actually come up with better designed next generation of probiotics that actually have a rational reason to be treating a particular disease rather than to make a particular food product. So I think we've learned a lot from the probiotic industry in terms of the limitations on what you can observe, as well as actually manufacturing organisms in high quality at large scale. But I think there's a lot more that can be improved upon.
The next question is, it seems that the elemental diet is beneficial in IBD, yet low in fiber. Why is this beneficial in the setting of IBD, whereas low fiber diets are generally considered less healthy?
Yes, and so the answer is that we don't know. What that means is that animal models aren't the complete answer, because everything in animal models would tell us that if you use that diet, you should actually get more disease. And what that means are humans are actually different. I right now favor the notion that there could be something else that we don't understand about these elemental diets that is having an immune regulatory effect, or it's very plausible that you are actually excluding something else that actually is bad in our current diet and whole food. Because we do know that if you start to eat whole food on those defined formula diets, you don't actually do as well. So I don't think fiber is the only answer. And that's just another reason why, in parallel from doing the things that I do in the laboratory at the wet bench, and I love mice, and that's how I do my research, it's actually fundamentally important to be thinking now at this stage of doing studies in human subjects in parallel, because we want to make sure that what we study in animal models ultimately, in some circumstances, will be relevant to human physiology.
And we have time for one more question, so I'll ask this question from an investigator who wants to know, in human microbiome studies with small sample sizes, around 10 patients per diet, how do you account for individual patient characteristics, such as age, gender, race, ethnicity, or comorbidities, that may drive association tests with microbiota and microbiota metabolites?
Yeah. And so, it's a really difficult thing in very small sample size. My only notion is that we try to make it as uniform as possible with strict inclusion exclusion criteria. Do you take probiotics? not using antibiotics, do the best that we can. The other thing that we actually do is we've been studying healthy human subjects because patients with disease tend to have a higher level of variability. And then the third thing that we try to do, well, the third thing we try to do is prospective studying, prospective sampling, so that you can actually look at, embrace that intersubject variability. So you may not see a difference cross-sectionally, but if you track somebody over time, they may have their own pattern. That's a very powerful statistical approach to be taking and something like that. And the final thing is that a lot of times in our studies, we actually introduce an intervention. And that intervention, thinking about it more dynamically than in a static type of way, actually provide a lot of significant information that, again, in our study, exceeded intersubject variability, even though we only had 10 individuals in each dietary intervention.
Thank you. We're at the top of the hour, and so this will conclude this grand rounds. But I once again want to thank Dr. Wu for a very comprehensive and insightful, thoughtful presentation on the gut microbiome in human health and disease. So thanks to all the attendees. Thank you, Dr. Wu.
Okay, thank you very much. I'm delighted to have given this talk. Bye-bye now.